Good afternoon, everyone. Thank you for joining us. My name is Marissa Waterman, and I'm the Marketing Director here at the Academy. Today's webinar is titled Understanding and Solving PFAS Pollution in Our Water and Wastewater Through a Holistic Approach. This is the final webinar in our PFAS series. Our Executive Director, Dr. Daniel Other, will serve as the moderator and will introduce our speaker today. During the presentation, you'll be able to submit a question by clicking on the Q&A icon on the bottom of your screen. We'll do our best to address each question at the end of the presentation. Before we get started, Dr. Other would like to say a few words. Dan, how are you doing?
I am fantastic. Good afternoon, Marissa, and hello to everyone online. And to those who'll be watching this recording sometime in the future, I'll say good morning, good afternoon, good evening. Welcome to one and all. As Marissa mentioned, this is the fourth and final webinar in a series of four with a focus on PFAS. And this series wouldn't have been possible without the leadership of Narapam H. and his team. Narapam was selected as a member of the Academy's 40 Under 40 award program in 2023. Our 40 Under 40 program recognizes exceptional rising stars who are making their mark on the profession. And as an Academy, we regularly reach out to this group of emerging leaders to solicit their ideas and invite them to contribute to the work of the Academy. This webinar series is an example of the type of leadership from these folks. So thanks to Narapam, and be sure to nominate exceptional emerging leaders for the prestigious 40 Under 40 recognition from the Academy. The goal of the Academy webinar program is simple: to support our members in your work. And as we begin today's webinar, I want to take a moment to offer some thanks. Specifically, I'd like to thank our patrons. Patrons are organizations who see tremendous value in the work of the Academy and provide financial support: $6,000 from utilities, $8,000 from private firms. And this helps us bring exceptional programming to our members. So please join me in thanking our patrons. And if you have an interest in knowing more about how your organization can become a patron, just let us know. And now let's get started. Today, I'm thrilled to join you to moderate what I know will be an exceptionally informative webinar entitled Understanding and Solving PFAS Pollution in Our Water and Wastewater Through a Holistic Approach. Now, that's a lot of words for a title, and that also means that we've got a lot of material to cover. Unlike our typical webinars where we listen to a single speaker, today we have a panel discussion, and I'm excited that we have three excellent presenters. Just as a little bit of a variation for how we're going to handle today, each person will give their individual presentation, and there'll be an opportunity for a little bit of Q&A, but that's mainly focused on targeted questions for that particular presentation. After we've heard from all three presenters, we'll then have a comprehensive panel discussion. So as the moderator, go ahead and put your questions in the QA function, and I'll decide how many of those we can answer right after a talk versus how many of those we save for the panel discussion at the end. But don't worry, we're not going to lose your questions. Our first speaker for our panel is Dr. Shannon Bartelt-Hunt. Dr. Bartelt is a licensed professional engineer, and she's board certified in environmental engineering by our Academy. Currently, Shannon is the Donald Volt Jr. and Nancy Keegan Chair and Professor in the Department of Civil and Environmental Engineering at the University of Nebraska-Lincoln. Shannon also holds a courtesy appointment in the Department of Environmental, Occupational, and Agricultural Health in the School of Public Health at the University of Nebraska Medical Center. Dr. Bartelt received her bachelor's in environmental engineering from Northwestern University, her master's and PhD in civil engineering with a specialization in environmental from the University of Virginia. As a researcher, she was recognized with the prestigious Career Award from the National Science Foundation. In 2015, she was recognized by our Academy as part of a group receiving the Grand Prize for University Research. Shannon is a fellow of the Association of Environmental Engineering and Science Professors, as well as the American Society of Civil Engineers, both sponsoring organizations of our Academy. Our second speaker is Dr. Olga Tuscu. Dr. Tuscu is an associate professor of environmental toxicology in the Department of Plant and Soil Sciences at the University of Kentucky. Olga received her bachelor's in biology in the Ukraine and then moved to the US, where she earned her doctorate at the University of Georgia. Dr. Tuscu has been supported by the National Science Foundation, the U.S. Environmental Protection Agency, and very interestingly, by a joint NSF EPA program with international research collaborations in the European Union. Since 2022, she's been serving as the U.S. chair for the U.S. EU Nano Environmental Health and Safety Ecotoxicology Community of Research. The goal of this is to bridge collaborations to further advance research on environmental health and safety of nanotechnology. And our third panelist today is Nirupam H. Dr. H is currently the Richard McNeil Associate Professor of Engineering in the Department of Civil and Environmental Engineering at the University of Nebraska-Lincoln. Nirupam obtained his doctorate in environmental engineering from the University of Texas at Austin, his master's in environmental engineering from the University of South Carolina, and Nirupam earned his bachelor's in chemical engineering from the Bangladesh University of Engineering and Technology. He's also very active on social media, and he and I regularly interact online. I encourage you to follow and interact with him there. Dr. H's research has been funded by the National Science Foundation, the National Institute of Environmental Health Sciences Superfund Research Program, the United States Department of Agriculture, the US EPA, the DoD CERTIP program, and the United States Geological Society. He's previously received the NSF Career Award, and as I mentioned in my introduction to this webinar, our Academy recognized him with a 40 Under 40 award in 2023. So this is a fantastic panel of exceptional scholars, and we are truly grateful for the opportunity to learn from them today. As a reminder, put your questions in the QA panel. For those questions that are immediately relevant to the presentation of each individual speaker, I'll do my best to have a brief Q&A after each presentation. But our goal here is to have a moderated panel discussion at the end. So I'll save those comprehensive questions for the end. Without further ado, I'm pleased to introduce Dr. Bartelt Hunt, who will be our first presenter. Shannon, the stage is yours. We're excited to hear from you.
Right. Thank you so much, Dan. I appreciate that warm welcome. And hello, everyone. I'm very pleased to talk to you today about a study that we've done relatively recently, looking at PFAS in wastewater and then also in municipal biosolids and after land application of those biosolids. So I didn't put in a lot of background information on PFAS because I feel like in the fourth webinar in this series, we generally have been talking a lot about PFAS, but I just wanted to highlight, I think, one of the most important motivations for why we're interested in PFAS, which is their human health risks. So exposure to PFAS, per and polyfluoroalkyl substances, has been associated with a number of important human health outcomes, including elevated cholesterol, alterations in immune system function, thyroid hormone disruption, fertility concerns, kidney and testicular cancer. And then very recently, some of you may have seen this paper that just came out a couple weeks ago that looked at PFAS occurrence and related that with the incidence of cancer for some very specialized type of cancers. And I just put one quote from the paper. So PFAS was associated with cancers in urinary, brain, leukemia, and soft tissues. And in females, PFAS was associated with thyroid, oral cavity, pharynx, and soft tissue cancer. And so again, I think this just really highlights the importance of why it is critical that we understand the movement of these compounds through the environment, their toxicity, and how we can treat and remove them. Also, many of you, I'm sure, are well aware of the drinking water regulations that have been relatively recently announced for PFAS. I did see that the proposed wastewater regulations were pulled back, and so there's a little bit of uncertainty there. But I think we know that the regulatory framework for PFAS will be something that continues to evolve in the near future. So in this study, and I have the citation for this paper, which is an open access paper, so something that you can read more about. We really wanted to do kind of a very broad study design to look at the occurrence of PFAS in wastewater, influent, effluent, and in the effluent mixing zones. And we did this using passive samplers, which I'll talk just a little bit about, some of the pros and cons of that approach. We also wanted to look at PFAS in municipal biosolids and then study what happens to the transport of those PFAS after land application of those biosolids onto crop fields that had an active crop growing. So really trying to look at the entire, I guess, life cycle from wastewater influent to biosolids and then land application, all within the same wastewater treatment plant. So this was just a single plant that we were evaluating, but trying to look at all those different compartments for PFAS. So this study was done in 2020, 2021, so and published in 2022. So we were not following the current EPA method with 40 PFAS, but following a more limited method that had 18 PFAS. But PFAS analysis was done using Xevo micro triple quad mass spec system. And like I mentioned before, for our aqueous samples, for our influent and effluent and mixing zone samples, we use passive samplers. So there's some benefits, I think, of passive sampling and what we have found in other environmental occurrence studies, that when we're doing this really broad kind of investigation, we can pick up very low levels of different contaminants using POSIS. There were some prior studies that we were able to quantify uptake of PFAS using these POSIS, and so we chose these passive samplers. What's nice about these is they can be put into the influent stream or effluent stream, and they will accumulate or aggregate the chemicals over a period of a couple weeks to a month. And so you're getting basically a time-weighted average concentration, not a point in time concentration. And so it allows sometimes those low level of compounds to accumulate over that exposure period. So kind of launching into the results, we've of the 18 PFAS in the method, we detected 14 of those PFAS in the wastewater samples. So this figure just is a plot of grams of total PFAS observed per day. We had two sampling periods. Sampling period 1, so you see some of those labels with a 1, those were partially out of the disinfection season, whereas sampling period 2 was entirely out of the disinfection season. So you see, I think, a couple of interesting things. One, we did see PFAS upstream of the discharge point. So all of the PFAS in the receiving stream was not coming solely from the wastewater treatment plant. We saw PFAS in the influent, but I think what is interesting is we saw an increase when we compared influent to effluent. And I think this has been determined in other studies as well, that there was actually higher amounts of PFAS in the wastewater effluent compared with the influent. Our hypothesis and that of others is that there are possible biodegradation of PFAS precursors in wastewater treatment processes that could be contributing to a higher amount of PFAS in the effluent compared to influent. And then certainly the highest levels we saw were in that effluent mixing zone, which I think just represents a contribution of the effluent PFAS combined with that background upstream amount that was already in the receiving stream. So essentially, message here is PFAS was in all of our samples, but we did see an increase in PFAS across the wastewater treatment processes. Looking at kind of the composition of the PFAS types in our different samples, so in influent, effluent, upstream, and in the effluent mixing zone, we did see some changes. So in the influent sample, it was predominantly long chain PFSAs or perfluorosulfonic acids. And we really saw a shift to a more balanced mix of different types of PFAS in the effluent. And so again, I think that just goes to the fact that there are changes occurring in the wastewater treatment processes that are going to affect both the amounts and also the types of PFAS that you're going to see in wastewater influents. And effluents. And then again, we also saw differences in different compositions in the upstream samples where there the short chain PFSAs dominated, and then the effluent where we saw the long chain PFSAs dominate. So again, just stressing that this is a very dynamic system. And as there are so many different PFAS, I think it's important to think about PFAS occurrence in different subgroups or subtypes. And so we did see differences in occurrence among those groups in these different types of samples. Moving over then to municipal biosolids. So we then took biosolids from that same treatment plant. Now the biosolids were not sampled exactly at the same time as the wastewater samples. So these aren't like time matched samples necessarily, but we did collect biosolids coming from the municipal biosolids program at this plant. And then we land applied those biosolids using an agronomic rate to some field plots, and then monitored those field plots over the months of September and October, the end of the growing season, where there was grain sorghum, a crop being grown on the plots. So the panel on the left just represents the mass of various types of PFAS that were detected in the biosolids and then also in the soil at the end of the land application study. And so the right plot just sums all of those PFAS. And so what you can see is certainly that there's a much higher loading of PFAS in the biosolids that were placed on the plot compared with what we detected in the soil at the end of the study period. We did detect PFAS in runoff, so that these plots were set up at an operating research farm at the University of Nebraska, and we had the ability to collect all of the runoff from that plot surface. And we did detect PFAS in runoff, so that does tell us that there's potential for transport of PFAS from land-applied biosolids. But we did see the concentrations decrease over our application period, which was August into September. Um, Clearly, there's a big difference. I think only about 3% of the PFAS from the biosolids was detected in the soil at the end of the study. So we absolutely need more information on either degradation pathway. So I assume that part of that PFAS loss is likely due to transformation or degradation away from the particular 18 types of PFAS in our method at the time, or transport of PFAS from land-applied biosolids. We did not detect PFAS in the crop, which was a grain sorghum. Other studies have shown PFAS uptake into plants. One of the issues with our study is we did not apply the PFAS at the beginning of the growing season. We didn't apply it until August, so it was only with the crop for a few months toward the end of the growing season. So that may be one of the reasons why we didn't see crop uptake. But with this particular crop, grain sorghum, we did not see PFAS in any of the internal parts of the crop. So some quick conclusions from this particular study. Wastewater treatment plants are likely a point source of PFAS to surface water. I will say that's a little, they're an accumulator of PFAS, right? They're not the source of PFAS, but they're taking wastes that contain PFAS and treating those wastes for other compounds and that are regulated. But also there may be processes, the wastewater processes may be creating PFAS, possibly from PFAS precursors. And because our study mirrored those of other studies that actually found higher concentrations in effluent compared to influent. And so again, that is not the treatment plants. Design purpose, right, to remove PFAS, but I think it's one of the unfortunate consequences of accepting all of this PFAS waste combined with the biological treatment processes is they may be creating some PFAS in that process. Also, we did detect significant levels of PFAS in the biosolids. So more work is needed to track PFAS degradation or transport in the environment after biosolids land application. And I think Nirupam will be speaking to this in a little bit, but that is one of our goals, is to really focus on the potential for PFAS to enter agricultural systems with this link from municipal biosolids. So with that, I will end my presentation and turn it over to Olga.
Well, I'm going to hop right in here right before we get to Olga. So a couple of specific questions that we had for you, Shannon. So you're talking about closing the mass balance. You're looking at degradation and precursors. Can you give us some examples of what those precursors were? Any particular names or off the top of your head or?
I can in a minute after I look them back up.
Okay,
Because
no problem.
yeah,
Nope.
we did have those. So let me see if I can pull those up.
Well,
We did.
they're probably in the reference, right? So.
They are, yeah. And there was a couple here, I can pull it up pretty quickly. There was a couple of very specific PFAS precursors that we were evaluating, and they were, yeah. So NET, FOSAA, so the N-ethyl perfluorooctane sulfonamidiacetic acid. Ooh, that's a mouthful.
That
And
is.
NME-FOSAA. So those two long chain PFAS, basically one is a 12 carbon chain and 17 fluorines, the other is a 11 carbon chain.
And so that's in the publication, so folks
Yes.
can look them
Yep.
up. Could those differences that you were seeing in PFAS be potentially due to differences in competition for other wastewater constituents? Like as you see things change in the wastewater, any speculation on that?
Yeah, I think that's a really good point. So I don't think our study was granular enough to really tease that out. But yes, I think that's definitely also a potential observation that needs to be explored more. I think our take-home message from kind of this broad swath and using passive samplers was just there is this potential for increase in PFAS concentration across the wastewater processes, and something that as environmental engineers, we're going to need to think about, and for plant managers as well, if they're, trying to request pre-treatment. I think we need to understand more about what some of those compounds might be that could lead to further PFAS loading in the treatment plant. So maybe those could be removed in pre-treatment.
And then last quick question for you. Can you give us just an insight into the nature of the facilities you were looking at? Large, small, municipal, industrial, all
Yeah.
in Nebraska, across the whole country, or?
Yeah, this was a largely municipal plant um, uh, in Nebraska. Yeah, I think trickling filter plant. Yeah.
Perfect. Thank you. Thank you so much, Shannon. Olga, we'll turn it over to you.
Thank you, Diane. Thank you for introduction. Shannon, thank you so much for your first part of this panel. And you actually saving time for me on some of my slides. So let me share it. Interesting. For some reason it doesn't allow me to share. Okay. Let's try that. Does it work?
We got you. Looks great. Thank you.
Awesome. So even though I am environmental toxicologist, I won't be talking about just toxicity of PFAS, but actually how can we implement sustainability approach, state by design approach in applications that we are developing for PFAS remediation, and also looking if we are degrading our PFAS in more toxic or less toxic compounds. And honestly, my background stems from working with nanomaterials and evaluating potential toxicity of nanomaterials once they release in the environment. And actually, after many years of looking at the effect, we eventually realizing that it's time to actually interact with engineers, trying to see if we can actually prevent the potentially toxic compounds, toxic nanomaterials released in the environment. And that's how I got involved into safety of nano-enabled applications, starting with PFAS remediation. And the next slide. Okay, so I won't go into details on this slide. Shannon, thank you already put in the motivation slide and describing the health effects and carcinogenicity and human are being exposed from the environment indirectly and directly through consumer products and from drinking water and so on. I want to bring you to the next slide that actually shows some concordance between shared health effects, between human health effects that are being observed based on epidemiological studies, and also the effects, toxic effects in wildlife, animals, and animal models. And there is definitely some concordance there, and such adverse effects, such as on immune system, developmental, reproductive, nervous. Also, one of the specific abilities of PFAS is to interact with lipids and disrupt lipid metabolism, resulting in the effect on liver, high cholesterol, and so on. And it's not surprising that that resulted in those stringent EPA's regulatory for maximum contaminant levels in drinking water. I don't think those were revoked yet, so those still stand, and at ways they're regulated. There are 5 compounds that regulated on individual level at 4 parts per trillion, at very small concentrations, barely detectable levels for all these compounds. And there are also 4 of those ones, including GNX, which is basically replacement for P4, are regulated as a mixture based on hazard index of 1. And when it's hazard index of 1 is meaning based on those ratios. And for example, this is concentration in the drinking water, and this is concentration at which health effects are not observed or not predicted to occur over a lifetime of exposure. So it's clear from Shannon talks, right, and from those regulatory maximum contaminant levels also, levels that we need to continue, basically, not only focusing on technologies that can remove PFAS, because then they end up in landfill a lot of times, right? But also to degrade. And there are a lot of different technologies right now currently under development, but they're not being used yet extensively. And that's what brings to this project. But also when we develop those applications, this is kind of like the diagram that shows how can we approach to such application in sustainable way. And there are different stages of development. I don't know why that. Different stages of development of the product. Started just a preliminary assessment where we can use already the materials, the results previously known about the toxicity of all potential materials involved, right? And predict the toxicity from that application if they are being released, any of the nanomaterials, for example, into the environment. And there are different those check marks between functionality and safety. And if it's not working at one of the stages and toxicity is prevailing, then going back and communicating to the engineers, and I have amazing collaborator on that, and trying to adjust basically the technology to see if we can make it as safe as possible. And this brings me to our collaborative projects with Isabel Escobar and Neuropalm Ike. So Isabel is at University of Kentucky and Neuropalm will be speaking next. And we were looking at actually developing of nanocomposite membranes for breaking down PFAS. And one of the potential 2D nanomaterials we are working with was black phosphorus or phosphorine. So when phosphorine is exfoliated from black phosphorus, and if you look at the, for example, band gap, right, diagram, the black phosphorus is actually takes that intermediate place between graphene that has 0 gap band and those transitional metal dichalcogenides that have fixed band gap. And black phosphorus has very tunable band gap, and depending on how many layers of exfoliated you can actually tune and enhance of different optical, electronic, and more important for us, for PFAS degradation, photocatalytic properties. So when you take that and want to implement that approach, safe by design approach in developing those applications, that involve evaluating all possible constituents included in the design before you can even test if your application is degrading PFAS into more or less toxic compounds. So this is just example, for example, of the membrane alone without even phosphorine. That's the filtration cells that we use. And we are evaluating toxicity of membranes alone, membranes with the phosphorine deposited on the membrane during fabrication. We also look at the toxicity of the phosphorine freestanding before even it incorporated. We need very sensitive endpoints for toxicity of PFAS alone. And finally, when we do our water spike with before and exposure through the after filtration through the membranes. So, but we expose not only to the filtrates, but also expose to the reverse filtrate. So we can see actually those breakdown products and whether they are causing toxicity. So this required to use of model organism, and one of the model organisms we are using is actually Cyanarhabditis elegans or nematode. It's a nematode that is being used frequently in different environmental toxicology to various chemicals, including PFAS and nanomaterials. And the advantage is just short generation cycle, 3 days from egg to adult, reproduction, but also the genetics. It's very known, annotated, a lot of different functional genomic tools such as mutants and fluorescent reporter strains that allowed us to go deeper and at more sensitive level with the endpoints that we select. And this is different example of different endpoints that can be looked with the nematodes. I know it's not PFAS toxicity yet. So when we just started working with the materials, this is our students, Luca and Usman, working together. Luca is my student and Usman is Isabel's student. And we just screen toxicity of the filtrates through the membranes without phosphorine. And what we started observing, we tested different pHs, that there is some variability in toxicity that affects actually toxicity and increased toxicity after filtration. And the filtration were recycling, kind of cycling filtrations. So we raised the question, what might be causing it? And this is actually the results for reproduction, and that's why that's decrease in reproduction at pH 7. So during fabrication of membrane, and you heard a lot about from DP about membranes last time, so there is a solvent that's being used during fabrication, which should be completely evaporated, but there is a potentially some possibilities that traces of that solvent and methyl pyrrolidone can be trapped in the pores. And when it's released during filtration through the membrane, it can cause that kind of effect. And what we did, we again, working with Usman together, Usman and Luca, they eventually came up with a modification in the fabrication and adding annealing step, which is the basically heated treatment after the membranes are fabricated, and that eliminated toxicity completely. So not even testing yet phosphorine membranes. Now we also look at the toxicity of phosphorine alone. Why do we need to know it? First, we evaluate mortality, and we see that the response is pretty at high concentration, 50 milligrams per liter. Those are really high. And those we needed just to see if we can go lower and look at the sublethal effects. And one of sublethal effects is reproduction, and we can see the effect at 2.2 milligrams per liter. So if there is any minimal release of phosphorine from the membrane during treatment, we want to make sure that it stayed below those levels, right? And after testing our phosphorine membranes, we did not see any significant toxicity from the filtrates. So now finally we are ready to test our phosphorine membranes, and we take our it's APA standard water that we use for media, spiked with PFOA in this case. and filtered through the membrane and looked at the toxicity of reverse filtrates. And this is example. So when we expose nematodes just to PFAS spike water alone, right, we have significant decrease, more than EC50, more than 50% decrease in reproduction. After we expose them to reverse filtrate without any additional treatment, or after UV treatment or oxygenation, we actually see that the reproduction returned back to control levels, which was really interesting. That suggested something happening with those breakdown products that actually making them to minimize that toxicity. And I won't go into details on XPS data, and there are additional characterization data that were done by Isabel's group. And that suggests that after filtration through the membrane, the P4 originally breaking down in more short-chained carbon fluorine compounds. We did not see free fluoride, so it's not demineralized completely. Also, this study prone us to search for more sensitive endpoint. What I didn't mention that when we tested before with wild type strain of nematodes with reproduction, we use 50 ppm, 50 milligrams per liter concentration. Very unrealistic for environment and what we are trying actually to break down, right? So now we are actually studying additional endpoints. We are looking at growth and at the behavior, such as locomotion behavior in C. elegans. And we are down below 1 ppm, below 1 milligram per liter for both PFOA and P4. And we can see, for example, significant increase in growth. This line, it's control line, and this is compared to control. So if we see significant increase, those are above the line for both. And for example, for growth, both mean worm area and worm length are increasing. And for behavior, and I just showed kind of like this a little bit as a picture here to explain that nematodes are moving sigmoidal movements and making that sigmoid movements, that's what accounts for wavelengths. And this is different tracks that they're making. So this is how track length is calculated. So it seems like they're moving, making more of those sigmoid movements, and also they're moving faster under exposure of PFOA and PFAS. And one more thing I mentioned before, that PFAS are known to disrupt lipid metabolism. It's been shown previously that it does affect the lipids in C. elegans, and we are looking at lipid accumulation in C. elegans. And also in response to P4, to PFOS, again, we are below 1 milligram per liter. We are trying to go to 0.4 milligram per liter, where we can see already increase in accumulation of lipids, which also makes sense why we see increase in the total growth, because previous study showed like risk for obesity in C. elegans specifically too. Now we need to get to more realistic scenario, because those compounds are not present alone in the environment, in drinking water or in wastewater. You saw from Shannon talk, they were screening 18 and detected 14. So now we are looking into mixture, and what is the between those, in this case, those two compounds, but also will be adding more. And if it's result in additive toxicity, synergistic toxicity, or potentially there is mitigation of toxicity. So far, those just really preliminary data that we tried the mixtures, and we can see that at 0.4 milligrams of both PFOA and PFOS, we don't see actually enhanced toxicity in the mixture. However, when we look at those effects, it needs to be a toxic concentration, and also different ratio of those compounds would affect toxicity. So with that, the take-home message. So I really, really hope that safety by design will be included in any application developments that we are conducting to break down PFAS to ensure we don't end up with more problems. And also that for PFAS are not don't have the typical acute toxicity. They don't cause acute toxicity. So standard endpoints are not working often for PFAS, and we need to be at as possible as close to environmentally relevant concentrations. So different endpoints need to be selected. And also very important, the mixture, right? PFAS mixture. And if the application is working, the new technologies are working to break down PFAS, are they breaking down to potentially more toxic or less toxic compounds? So with that, I would like to thank all my collaborators and students in the lab. And if there are any questions.
Olga, that's fantastic. Thank you so much. So just a couple of quick questions specifically related to yours, right? So one of the things that you highlighted was that you had these individual compounds or then these kind of simple mixtures as compared to kind of the real world where there's lots of things there. How should we get our mind around taking the kind of the toxicology information to understanding the epidemiology endpoints that we're using to set these kind of regulatory levels? Are we, is there room for improvement there so that we're not having to spend as much money to try to treat quite as much as we're trying to do with these very, very low MCL kinds of levels?
So the MCL level, just to kind of to clarify a little bit there, based on the response to vaccine. So it's decrease in antibodies, and I think it's based on the depth vaccine. And also, there is the suggestion from the human epidemiology study that basically there is decreased immune response to also viruses, for example, infection diseases, right? So that's, it is really low, right? And epidemiological studies, a lot of them are also done with populations that are close to the source. So the level more contaminated to see those responses to. So how do we translate? That's a equation, good rule equation. How do you correlate between epidemiology study and the animal model studies? Because epidemiology looking at the patterns of diseases, right? And toxicology is actually looking at the adverse health effects in specific organisms.
Sure.
So I think that one of the references I posted to the kind of like that they look at wildlife animals and then the human studies, and if there is any correlation, and they have like really, really nice tables to really compare, like, but it's so organism specific, species specific. It's just mind-blowing, and then what the organisms are exposed to the environment, it's not the same as humans are exposed to. It's totally different. So there is some concordance, and I think having those animal models does help. For example, with C. elegans, it seems to be okay, you're using really basic nematode, really small one, right? Is there a conservation? Yeah, there is quite a bit of conservation between biological processes and C. elegans, maybe more simplified, and in mammals. And with lipids, there is 302 lipids in nematodes that are homologous to lipids in humans. So there are definitely those conservation. I'm not suggesting that just based on the nematode study, we can say, okay, it's safe. But we can at least say what it's not safe already to start with, right? When we are implementing those toxicity testing for applications.
So a couple of specific questions for you that also came in is about bioaccumulation. Was that something that we know a whole lot about yet, or not really, in terms of mechanism?
We do. C. elegans not very good model for bioaccumulation because of the size. It's true, but from the other animal model studies that PFAS, they are prone to bioaccumulation. They are accumulating, not exactly in fatty tissues, but I mean, the other study do show it. And
are there
because they're persistent, they stay.
They're persistent, so they're there. Okay, so not necessarily bioaccumulation, but we find them everywhere, perhaps, maybe.
Well, yeah, and once they're being taken in, they're not being eliminated.
Okay.
I mean,
so they will accumulate in that way. Okay. And then I guess one last one before, for you, because I'm trying to get a couple of questions for each person. So a specific one, do you note any trends in what we might say like the toxicity among the traits of the PFAS, the shorter versus the longer versus are there trends that we're seeing starting to emerge in these different compounds?
Yes, actually, so that's why like before, right, was replaced with Genex. It's not in production anymore before, but we still have it in the environment. And Genex is C6 and before C8 compound, right? And we know, for example, that the short length compounds in general do seem to be less toxic.
In
But
general.
in general, if you look within the group of those short chain, actually Gene-X would be the one that is prevailing and it's still causing toxicity. So it really depends on which class you're looking within. So you need to be careful. So you cannot make very general, and some of them have very specific mode too, of toxicity that is not necessarily compatible. Like if they have the same mode of action for toxicity, then we expect them to have additive effects. They may enhance toxicity. But what if they have different mode of actions? What do we have now?
Correct. Correct.
Yeah, it's definitely not a simple answer to that question.
No, that's fantastic. Thank you so much, Olga. We appreciate it. Narapam, we're going to turn it over to you, my friend.
Okay. Let's see. Whether I have the presentation. Okay. Can you see my screen?
Yes.
Okay. So I'll go with this. So thank you, Dan, for making this happen. And so before, thank you, Olga and Shannon, for giving this wonderful intro and all the discussions about that. And I will tie back to their talk, but I will first introduce my lab, IH Laboratory for Environment, Nanotechnology, and Sustainability. We focus because the work that we do, I don't do that myself, my students do, and we work as a team. So right now I have a bunch of PhD students and a postdoc and an undergrad student. Most of the work that I'll be talking about has been done by Dr. Arvid Masood, who is now working in Intel Corporation, and then currently by like Mr. Arafat Ali. So, but we do take pride in our group, and this projects on mostly focused on PFAS treatment has been supported by several federal agencies and with collaboration from several folks and colleagues from across the country, including UNL and UKY, like Olga already talked about our collaborative project with Olga and Isabel. So with that, I will go into the discussion of PFAS concentration and how we are trying to use nanotechnology for destroying PFAS in wastewater systems. So I'll not go too much detail in the specifics of what is the limits because Olga already mentioned the maximum concentration limits from EPA in drinking water. But the main challenges are with those PFAS concentration limits is that they are very low, that has been set. The second part is that the conventional treatment processes for either for wastewater or drinking water treatment, these treatment processes are not as effective. So we are looking at like $200 billion to $1 trillion capital cost for water utilities, which is a huge problem. So what at the same time, if we look at the PFAS treatment approaches, there are actually a lot of work going on from different technological point of view, like trying to look at granular activated carbon for treatment technologies. This is really nice summary. I forgot exactly where I took it from, but I believe it was one of the companies that they put together this short summary. So they identified like there are some mature technologies like ion exchange resin, nanofiltration membrane, reverse osmosis. Obviously, if you try to take out PFAS from water using nanofiltration membrane or reverse osmosis, they are very, very tiny membranes, so they can possibly take out most of the PFAS. These are in trace contaminant, but the issue is because these PFAS are in trace contamination, trace concentration, there is a lot of water flowing through. So reverse osmosis and nanofiltration, there is a lot of energy cost there. Unexchanged resin is honestly a good option for taking out many of the PFAS, either short chain and long chain, because you can, there are some recent research going on in looking at how you can modulate the unexchanged resins in the unexchanged process to target short chain PFAS compared to long chain PFAS too. Granular activated carbon is really, really good at taking out um, some of the long chain PFAS like PFOA and PFOS, but they don't really work well for short chain because of the hydrophobicity versus hydrophilicity issues in case of short chain. One of the major things to think about, like when we talk about PFAS treatment, we are not only talking about concern about PFAS separation from water, because the technologies that I just mentioned, they separate the PFAS from water, but they don't really degrade them. The main issue for their toxicity and persistence of PFAS is their carbon fluorine bond, strong carbon fluorine bond. So one of the major way to think about is how we can develop technologies that can destroy this carbon fluorine bond. So we need uh, improved efficiency. So if you look at the other processes like thermal process, electrochemical process, plasma process, photocatalysis, there are some technologies that are coming out mostly in plasma, electrochemical, photocatalytic. These are some of the processes that are showing some evidence of destruction of PFAS, but there are a lot more work to do. The unfortunate thing is that biological treatment process really is not working that well. And that is very unfortunate because biological treatment would be one of the like most cost-effective approach to treat PFAS. Okay, so why am I talking about this? I'm talking about this because I want to show our work and what we are trying to do to achieve PFAS separation and treatment. So our main focus has been we are using nanomaterials and nanotechnology for taking out PFAS. Olga mentioned some of it, our work together, but I'll give an overview of what we are trying to do. So nanomaterials, if you're not familiar, nanomaterials are very small solid materials in atomic scale or nanoscale that can take that we can use to treat water. They are applied mostly in electronics. We get all these smartphones because of the chips that we can make in very small scale. Okay, so we are now using these materials to take out PFAS. So what we have done in our first work that came out in 2021, we tried a nanocomposite. We say nanocomposite or nanohybrid material where we are combining two different materials. Say for in the right-hand side figure, you can see in the bottom, there is this carbon-based sheet, the black carbon-based sheet, graphene sheets, which is a two-dimensional graphene carbon-based material that can help absorb contaminants on its surface because it has a carbon backbone. But also it has some functional groups. We are using reduced graphene oxide in that sense. What it means that it is carbon, but it has some carboxyl, hydroxyl functional groups on the surface too. So that can also interact with some PFAS based on electrostatic interaction too. So that can help absorb PFAS on the surface. But at the same time, we are combining this carbon-based material with metal catalysts. In this case, iron or nano-zero valent iron nanoparticles. The reason we do that because iron nanoparticles are very good at uh, in catalyzing hydrogen peroxide to create these reactive oxygen species that can potentially degrade a lot of organic contaminants. So we tried to see if this is going to work for our treatment process. So either can we absorb these chemicals on the surface. So what we did, we took three different materials: iron nanoparticles, or NZVI, graphene oxide nanosheets, and then we combined graphene and iron together to see if this hybridization or combined approach will be better or worse for PFAS treatment. On the right-hand side, you will see these short chains. We took four different types of PFAS. The 5 carbon chain PFAS, which are short chain PFAS, and then 8 carbon chains PFOA and PFOS, with, like, you can see that there are differences in functional groups at the end too, to see if these nanomaterials can take out PFAS through adsorption or oxidation processes here. So how we did them? So we basically take PFAS solutions in vials, and then we dose them with different nanomaterials, depending on which nanomaterial we are looking at. And then we shake them for some time. And at different time points, we take out supernatant and centrifuge them and see how much PFAS has been absorbed or degraded, how much or degraded over time. So we use liquid chromatography mass spectroscopy to analyze the PFAS concentration. So what did we find? What we found is, okay, we took four different PFAS. So if you look at the left graph, and the writing is on the top, but on the left hand side graph, there are different colors. But if you just look at these short chain versus long chain, on the right hand side, these short chains like long chains, PFOS and PFOA, they have really high hydrophobicity, 5.1 and 5.4, compared to shorter chain PFAS, which means that these shorter chain, our nanomaterials, are more effective in removing longer chain PFAS than shorter chain PFAS. The next thing I would say that look at the colors. Like, there are so many colors, like gray, red, and green. The gray one is just the graphene, the red one is just the iron, and the green one is the nanohybrid NaH we have put. So if you look at them in each of these, in case of each of these compounds, let's just focus on PFOS. In this case, you will see that the green bars are mostly at the top. Yes, there are some differences. I'll go into that. So the green bars without the hash lines, you can just compare them. You see graphene, there is a 70% removal of PFAS, with iron, 60% removal, but with nanohybrid, we have more than 80% or 90% removal. So one other thing we have done, we have put hydrogen peroxide with them, where we have these dashed lines in these bars. So if you look at them, you see the highest one is actually coming from when we have hydrogen peroxide. So with nanohybrid. So we are looking at this, and we are thinking, okay, this is really good. And when we look at the kinetics, if we see just the nanohybrid itself to remove PFAS, we see that nanohybrid itself, when they are only doing the adsorption process, we see within 5 minutes, we see 50% removal. But if we use hydrogen peroxide with it, which can create reactive oxygen species on the surface because of the catalyst, we can see a 90% removal within the 5 minutes. So this is really, there are three things that are happening. Long chain PFAS remove more than short chain PFAS. The nanohybrids work better than the parent materials, and the hydrogen peroxide catalysis can really remove more PFAS and faster than adsorption process. But is really PFAS degradation happening? So we have colleague in this project, Dr. Diana Aga from University at Buffalo. Her group specializes on high resolution mass spectroscopy to identify degradation products from different organic contaminants. So what we found is that we have found that several of these PFAS we found several shorter chain PFAS, PFPS, PFHXS, all these shorter chain PFAS are produced. And with high resolution mass spec, what we found is that there are interestingly several PFAS complex. These are say 6 or 7 carbon chain PFAS, not like really small carbon chain. And at the same time, we found very little amount of fluoride. So the good news is that it's not only adsorption process, there are some degradation process. The not so good news is, although there are some degradation, but they are not degrading totally. There is a very little amount of mineralization and carbon fluorine bond breakage. There are they are coming from 6 to 4 carbon chain. There is partial mineralization, but not total mineralization. So we have to do more work. Okay, so at the same time, the next project that we did, we wanted to understand if this degradation process or adsorption process is going to be affected by water chemistry. This just came out last year, and we have seen that different water chemistry, understanding the effect of water chemistry is very important because our wastewater or groundwater, drinking water treatment processes are going to deal with different pH level, different natural amount of natural organic matter, different dosage of or distribution of PFAS concentrations, and can we optimize those processes with different oxidants? I'm not going to talk more detail on it, but the main idea is if we have our processes work better in low pH, so we need to really control the pH when we are doing the PFAS treatment, and then probably go back up at pH 7, around 7 to distribute the water after the treatment. So there are some considerations like that. How do you optimize the water treatment systems for these conditions? So with that, we have seen some partial mineralization. So we wanted to improve these processes. So now we are looking at a different catalyst, photocatalyst, titanium dioxide. Titanium dioxide is a very well-known photocatalyst. These are used actually in paint for degrading any biofilm or mold formation, preventing any mold formation, because they also, under sunlight, under UV radiation, they can create redox oxygen species and highly reactive species that will interact with organic materials. So we are now testing different types of PFAS. I'm showing that one PFAS, we have seen that over time, we can degrade PFOA quite a bit, like goes to almost 0. And then we have seen quite a bit of fluoride generation with this. I have more data. I'm not showing all the data, but we have tried with 8 different PFASs, and most of them show very good results. And we have seen much better mineralization with these materials, graphene titania materials, instead of graphene iron materials. So these are different approaches we are taking to make these separate PFAS and destroy PFAS too. So I'll go in another side. So while these fundamental experiments we are doing, this is really encouraging, but most of these nanomaterials come as powder. So applying them in treatment system can be very challenging. So this is a project that actually I presented it last year, details about this project last year, that we are using 3D printing to create more like a filtration device to use these catalytic materials to design different porous materials for filtration and destruction of PFAS at the same time. So this is funded by NSF Career Award, and recently we have got funded through US EPA to kind of combine these nano catalysts. Our catalyst is iron or titanium-based catalysts with nanosensors for field deployment. So this is more applied work we want to see in case of groundwater or wastewater systems. So we have colleagues in Clarkson University and Stevens Institute of Technology in New Jersey. They specialize on developing like very small scale sensors, nanosensors that can be deployed in field. So we are trying to combine their module with our module, treatment module, to see can we develop like simultaneous detection and treatment technology for PFAS that can be field deployable. So this is very interesting because PFAS analysis in field is really challenging. So if we can do that, that will solve a major problem for detection, but also degradation. The processes that we are setting up in the lab, are they applicable in the field? So with that, like these are most of the projects that we are working on. And then why we are talking about needing for holistic process. Okay, so Shannon really talked about how the issues of PFAS in our wastewater going into the biosolid and firm, right? And that really is one of the major issues. So I'm coming out from the nano side and I'm talking more about why we think there is a collaborative approach needed for all of us together. So we are developing, like I am developing treatment technology. Uh, Shannon is looking at PFAS fate and transport and occurrence, and Olga is looking at toxicity, but all of them are important with each other because these problems are not one at a time. So if we look at the problem, municipal wastewater treatment facilities need to be looked at to understand how PFAS is in water, wastewater, and biosolids. How much of them go into the environment through biosolid application. But at the same time, when we are trying to determine wastewater treatment approaches with nanomaterials or nanotechnologies, at the same time, we have to understand if these nanomaterials are going to cause a problem, or the PFAS degradation products that we have seen that not fully mineralized, some partial degradation has happened, are they going to cause a problem? But overall, PFAS can enter our agricultural system, can exist in our food system. So when we are looking at this, we are thinking about Nebraska, which is an agricultural state. Kentucky has a lot of, like most of our states in United States, many of our agricultural states face similar situation. So overall, the impact of water agriculture, impact of PFAS contamination in water agriculture, food, and economy is very, very going to be astronomical. So thinking about that, like we need to think about PFAS occurrence, distribution, PFAS toxicity, treatment process, if these treatment processes are effective or not, uh, and how they impact our agricultural systems, how they impact our drinking water, how they impact our private well water in rural areas. So we try to think about holistic approach. So what we tried to do is last September at UNL, we organized the Great Plains PFAS Initiative Symposium and Workshop. So we brought in, you can see here, like if you look very closely, there is Olga back there and Shannon back there. There is Isabel here. So we brought in all these different states and like universities from 7 different states, uh, 7 state, 9 university, more than 60 participants to understand the PFAS pollution in human health, environmental health, and agricultural communities, and how we can combine our academic understanding, research activities, industry, and utility together, along with state and federal agency to address this problem from a holistic point of view. So we are trying to establish a consortium to understand the PFAS pollution problem and try to mitigate it. But at the same time, can we educate the communities about the risk and give them hope that we are working on it and solve this problem for everyone, not leaving behind anyone. Okay. With that, I will thank you all for listening, and I will hand it over to Dan from here.
Fantastic, Norpo. We've got a couple of specific questions for you, and then we'll open up a little bit broader for some panel discussion. So specific question for you. Generally, when we go to shorter chain PFAS, when we go to degradation products, not destruction products, but degradation products, are they typically less toxic, better? I guess some people are thinking, right, we're thinking dry cleaner fluid and we end up in vinyl chloride, and that was actually a worse outcome than what we had started with TCE. So how about with PFAS? Is some destruction better or worse, or do we not know?
That's the last one. We don't know. We really need to do the work. So we haven't done that like degradation product assessment, toxicity assessment. That actually reminds me, probably I should talk to Olga that, oh, can we send some samples to you to start doing the toxicity tests? But that's why we are thinking about it. Like we should really need like think about looking at every, like not every degradation product, but the different types of degradation product, if we can look at if there is any correlation between which transformation products we are getting and toxicity relationships. Some are probably less toxic, some are probably high, and can we tailor our treatment technologies uh, to like more reduced toxicity products, byproducts? Yeah,
Perfect.
if
Makes
not
sense.
mineralization. Yeah.
Now, when folks are conceptualizing this idea of nanoparticles and you're talking about sorption, you're talking about destruction, from a kind of a rudimentary kinetic standpoint, are you talking about kinetics in solution or are you talking about kinetics on surfaces? How are we visualizing, conceptualizing these nanoparticles, and then, they're got to be attached to something so you can, capture them back. So how do the engineers conceptualize this? Surface
That's a great
reactions or soluble reactions?
that's a great question, because what we are figuring out is that it has both components. So, like for the treatment, like for the treatment for iron-based system, so we are now looking at how much. So what we did first, we looked at just how much degradation product is in, what are the degradation product in the solution phase. Okay, so even though the reactions are in those degradation products are in solution phase, but they may have happened very close to the surface. So these are surface initiated reactions, but the reactions can be near surface or on the surface. Okay,
Okay.
so there can be two parts. And in other side, what we tried to do, we took out the particles themselves after we did the treatment, and we dissolved them, and we analyzed the surface, like any PFAS, are there any PFAS on the surface or degradation products on the surface? So we have found some degradation products on the surface too. So there are multiple things going on. There are some solution phase interactions going on, and then surface phase. Even after absorption on the surface, there are some reactions going on the surface. Yeah, it's
Okay.
both.
Okay. Both. And if we're talking about these applications that you're talking about, right? So I imagine on one end, you've got some pre-treatment and some finished drinking water, and you're just doing a polishing step. And that's kind of, you've got a really good input stream, and you're just making sure there's no PFAS left. And then you might move to kind of raw water. And on the other end, you might have Shannon's biosolids or soils, but irrigation water is going to fall somewhere in that spectrum. So when you're talking about these technologies that you're developing, are you focused primarily in that finished water, the polishing step, or do you see these being able to work in quote unquote dirty environments like raw water or irrigation, or all the way over to wastewater treatment finishing or biosolids destruction? Where does these technologies you're talking about fall in that continuum?
Yeah, that's another great point, because I think it depends on the water quality in every, like it will be case by case. If we have too dirty water or we have a lot of NOM, probably most effectively they will be most effective in the polishing step. Like what we are not sending it to reverse osmosis, we are doing it in there. And the other reason to do it is to reduce the cost too, because like there are some technologies, initial membrane technologies, that can take out many of the PFAS, and you could not only PFAS, but also other contaminants together too. So is that good or bad? And then you can have a disposal system or like post-treatment system for those. So then whatever is getting cleaner, but PFAS is not degraded in or removed, we can take care of that. But there are some like when we are testing like um, different water chemistry effects, we are also trying to think about, could we tune it to go to raw water or more complex
Mhm.
systems? There would be a need for that too. But I don't, like at this point, I think they are better for polishing and like taking out trace contaminant, then going into the much more complex system. Yeah.
Okay, fair enough, fair enough. And maybe if we pull down the screen share, maybe we'll have all three panelists come up, and maybe we'll have a little discussion here. So if folks have broader questions, feel free to put those in. I've got a couple that we can maybe start with. And so kind of a big high level question, and anyone can feel free to jump in and answer this. But if we're kind of going back and conceptualizing, right? We're thinking like on the dirty side, the Clean Water Act, and we're thinking about like on the clean side, the Safe Drinking Water Act, right? So we've got these kind of regulations that have been with us for 50 years, and they were designed in an era when we were thinking about stuff like organics impacting oxygen in streams, or maybe some nutrients, or maybe some fecal material causing people to get diarrhea. So that's kind of where we're at on the wastewater side. And then we get to metals and we get to other things like that. On the drinking water side, we've got this kind of never-ending MCL. Let's get this compound, let's look at this compound. And so it's kind of the regulation is set up that we will always be adding another thing to go after. So I'm kind of curious from the perspective of, we've got microplastics we're worried about. We've got antibiotics we're worried about. We've got the classic nitrogen and phosphorus that's still causing huge amounts of eutrophication. We've got limitations on total water. So we're thinking about, indirect potable reuse and all of these. Where do you feel PFAS at a high level fits into this constellation of, we've got, 10 or 20 trillion-dollar problems that we need to solve, and they don't necessarily all get solved the same way. So how does PFAS fit into that constellation of all the things that we've got on our plate to deal with?
That's a really great question, Dan, and I'm not sure that I have a great answer, but I do think that when we think about treatment or occurrence or toxicity, we definitely need to start thinking about techniques and strategies that will address multiple contaminants, because obviously none of these compounds that you've mentioned are going to exist in isolation. So they're all going to be in a mixture. And so I think, yes, we need to start looking at things more holistically and addressing solutions that maybe can address multiple different types of contaminants. I think also, obviously, there's going to be, most of the work is obviously regulation driven. So I think it's going to be interesting too with drinking water regulation, but then subsequently, if we have delays on regulation and waste, that, and some of the things you mentioned are completely unregulated, like microplastics. So I think there's also that kind of practical element that at some level we probably need to focus on, working within those regulations, but then also addressing and seeing how those solutions can affect unregulated contaminants as well.
Fantastic. Olga, thoughts on that? It's a tricky question, right? But it's the very practical one, right? So
no, it's really coming to more realistic scenarios, right? What we are dealing with real world samples. For toxicity point of view, yeah, I think it's really critical. Not a lot of people want to go there because it gets much more complex once you start adding contaminants on top of another, right? But we got to think about co-contaminants and how they interact with PFAS and how it affects toxicity, and whether our treatment technology can potentially remove both. Is it possible to kind of functionalize it in the way that it's working not just for PFAS? But yes, the mixture toxicity is huge. And also, like, I'm using C. elegans. I cannot put effluent and test toxicity of effluent on C. elegans. Kills on the spot. But we have other model organisms such as Daphnia that is specifically for effluent toxicity testing. So there are ways of expand to more mixture toxicity. And even if we can go to simulated, to the real world sample yet, we can try with simulations. And there are some studies already testing different mixtures that are present in the wastewater, right? But simulated in the kind of like more clean and see how toxicity varies. But yeah, very, very complex area. I agree.
Very much so.
Yeah,
Nirupam,
actually,
thoughts on that?
yeah. So in our workshop, actually, this came out at the end. Like now we are thinking about more about one water, like
Mhm.
approach, like thinking like we shouldn't think about only one, how we can think. But it's difficult, like, like difficult because we each have our own group. We are doing one thing, and then you try to do all these things. But from the other point, like we are working on nanotechnology, and the question that you asked me first, like, is it polishing? Is it this or that? That is really where the question would be, like in terms of treatment. I think even for PFAS, like when we talk about treatment technology, we shouldn't talk about treatment technology. Oh, is this best or the other? Like more about like how we can put technologies together. And I think PFAS is such a problem that problem where you need treatment train approach instead of treatment technology approach. Like, oh, this is the one thing. Like for disinfection, we do chlorine addition. That's it, right? You don't have to worry about or some UV, right? But here, PFAS is such a complex thing that you need to think about treatment train approach instead of like the NIH project we had. We started with the initial iron-based project I showed. I didn't talk about the later part of it. The idea of that was, can we initially break down or transform partially these um, PFAS compounds so that the bonds are weakened, and then we send it to biological treatment
Okay.
process. So it was honestly not targeted with drinking water treatment. It was targeted with wastewater treatment. So now we are doing some experiments. We are getting some results which show, like the paper is getting written, but which show that, okay, we are not really improving the treatment efficiency as much as we have thought, but we are improving the reaction kinetics in the biological treatment process. So we are shortening the HRT. So like, which is encouraging. So I think that approach and like thinking about if there are multiple pollutants, which one we take out first, which one next. So that might be a way to think more treatment train approach than anything else. Yeah.
Perfect, perfect. We had a question that came in, and it might be outside of the scope of you folks, but you might have heard of it, or maybe it's not outside the scope. But part of it is the pollution prevention side, right? Like, and so yeah, there's these, the PFAS that we'll be using so that we can have, no stain fabrics and Teflon coated pots and pans. So we can imagine those kind of places that maybe we can get rid of them, right? But some of these applications like firefighting foam in particular places. So are we at a stage where we're not making the problem any worse right now, or do we still have a worsening problem while we're still trying to think about how to solve it? Can you, yeah, wasn't one of the specific things we talked about today, but it was a question that came in, and I think it's kind of a fair question. Do we still have problems because we're still making it and using it in places?
I will go first. So what I have, there are two examples I will give. I think firefighting foams have been quite, like they have quite evolved, and there are firefighting foams that are now used that doesn't have PFAS. But is this less toxic or more? That's a different question. So, okay. But there has been some directives from administration or government to replace these firefighting foams which contains PFAS. The issue with that, like this is where the community engagement is so important, is that many of the fire stations that are in like smaller cities or rural areas, they don't really know that. They're still using that. And there is not much of communication or like extensive effort in that. Because these firefighting foam efforts are coming mostly from Department of Defense and less from the civil side of it. Okay.
Sure.
The second part, I'll give another example in case of semiconductor industry. So semiconductor industry, there has been a huge push from the last year, like early last year, that PFAS is used in all semiconductor manufacturing. And they have they are trying to take out PFAS, but how do you define PFAS? The issue is that for all their production, they need PFAS, some sort of PFAS, fluorinated compounds. So they have moved away from PFOA and PFOS using, and they were told initially that the other PFASs are fine, C1 to C4, but now we have broadened the definition. So now they are trying to come up with, there is a huge proposal call that has gone through to understand what are how much concentrations are there in semiconductor industry, what they can do to replace them, if they cannot replace them, how can they have their own treatment technologies on site to deal with the issue so that they are not polluting the general water systems. Okay, so these are the two examples I know. Shannon, you were saying something.
Oh, no, I was just gonna say, yeah, it's not my direct area, but in the PFAS workshop that you mentioned near Palm, I think um EPA speakers mentioned that some of the replacement compounds for the firefighting foams have increased toxicity. So I don't think we might be shifting away from PFAS, but we may not be solving the problem entirely with a non-toxic or less toxic solution.
Fair enough.
Yeah, but the good thing would be even if they're toxic, if they're less persistent
That's what I was gonna
and
add.
biodegradable, that would be probably good. I mean, overarching.
Well, Olga,
Like,
that's kind of where I was going to go with a question for you. Like, if you can elaborate on that one, but also speak to, because we're the American Academy for Environmental Engineering and Science, right? So there's also that idea that what are we doing to protect the environment? You know, if you think back to the ban on DDT, sure, you think about human health, but you also think about environmental problems as well, right? And so great that we don't have PFAS in water humans consume, but if it's out there, so, you know, how do we understand PFAS in a natural environment as well? Can you speak to that some?
And natural environment. So that bring back maybe examples from nanomaterials. That's when we just started studying nanomaterials, what is it in pristine form, that they are as synthesized. Then we realized that it's not realistic, right? They get modified in the environment. So we started modifying them. And eventually we moved into mesocosm studies, where you actually have more realistic environment. So maybe, and I know Tiffany Messer, right? Like, for example, studying the transfer of PFAS in wetlands. So she is more in realistic environment. And I don't remember if Jason White is looking at also at the PFAS in agricultural plants. So if you really want to know how something behaves in the environment, even greenhouse studies sometimes is not exactly gives you a complete picture. You need to go to the more field studies. If it's animals, it's mesocosm studies. But that gets very, very complex because you get the trophical accumulation, right? And you have different level of the food web. So, but do we want to go there? Are we kind of at the point where are PFAS? Do we want just to step away from PFAS and find good alternative? But yeah, what Shannon mentioned about the talk, you know, Mocha talk by Mocha from EPA, he was talking about F3, basically PFAS, and all of that look like resulted in acute toxicity, but they're persistent, like biodegradation basically seems to be, they were still continuing their studies, but that's where they were pointing. Okay, it might have caused acute toxicity, but if they degrade fast enough, then we might be better off than using PFAS at half-life, and bioaccumulation point is much higher, right? That's why they accumulate, because their half-life is much longer. So, but yeah, we don't want to create something else, but it feels like with all the big push on PFAS, eventually industry will be like, if there is any way we can just eliminate PFAS completely.
No, it makes sense. It's a tough balance, right? Because again, as engineers and scientists, we're imagining solutions we've had in the past from a policy that match with technology, right? Like the surface water treatment rule. Let's have a package of technology that will get rid of most of the things we're worried about, right? And that's kind of where my question was motivated with the microplastics and the antibiotics and the synthetic estrogens and the PFAS. And so we want to keep people from being exposed to all of these things. So is there a surface water treatment rule analogy for all of these pollutants, right? I'm sure, Shannon, you can imagine where I'm going as an engineer thinking about that, right? We're not going to have these specialized treatment trains for every single municipality and every single small, town and hamlet. So Shannon, a specific question that came in somewhat related to yours is there any correlation with Class A, Class B biosolids? Are you seeing anything that relates to where we would think about with land applications or incineration or other things there?
Yeah, that's a good question. The I'm sure there are differences. We were working with Class B biosolids. I think um, clearly, I think higher levels of treatment I would expect would result in lower PFAS loading. And I think there are definitely things where we could look at more treatment of solids in addition to wastewater. So that's probably um, another active area where we need more study is not just looking at treatment of the aqueous phases, but we also need more treatment of the solid materials before we do things like land apply. So
Yeah, it's a tricky one to get at. There were some great questions that had come in with folks. Some of them are kind of, very specific ones, right? There was a question looking at some EPA data, looking at foam fractionation and comparing PFAS. Does anybody want to take on that question? I know we discussed that some in our last webinar with the foam fractionation, but different because I mean part of this is let's separate them out of the water and then let's worry about treating them or destroying them separately. Others are, Neuropam, to your point, let's separate and treat at the same time, but maybe that's only as a polishing step. What's been neat about these, the four different, the three different webinars we've had up to this point is that we've had people come in and talk about it from each of these perspectives, right? And so now today's kind of putting it all together, and that's where it's neat to talk about these high level, but I'm getting into some of these kind of specific questions as well. So Narapam, I don't know
Yeah.
if you had any comments on the foam fractionation part or
No,
not.
I think this is a good point because like when we do treatment technology design, like there is always this question like, oh, you are putting so much energy to do something. And like when we studied these treatment processes, we are testing our lab scale testing are happening at 400 ppb. And there is always this question, okay, your environmental concentrations are in parts per trillion. What are you doing? And then our main logic is, oh, there will be technologies like foam fractionation or membrane processes that will create these concentrates. And that's where we will put this like technology, not necessarily. And I think although I'm talking about separation and destruction together, but there, even before that, if these technologies are to be used, if there is a separation technology that takes out like from water where there is low concentration, because if you think about the EPA limit, it's 4 parts per trillion, which is crazy, right? So, like many water systems, I have seen like when people are saying like, oh, in water system, if you get 400 parts per trillion, that's a high concentration of PFAS. But it's very difficult to get a treatment technology that can go at that level and be efficient and cost effective. So having a technology to concentrate is going to be useful for any destruction technology development. Like if there is a pre-treatment which can do concentration, and then it comes to the next step, that will be also very, very useful too. So I think there is a potential for using these technologies.
Fair enough. Fair enough. And then Olga, there was a question in here that's a little bit maybe more in your wheelhouse. There were a lot of slides talking about PFAS and human health effects. And, we've got these very, very low numbers that we're trying to get to. Do we really, I guess maybe a way to word it is, do we really need to do that given all the other things that, whether it's vaping or obesity epidemic or diabetes or, is this really kind of the thing that we need to be chasing after when we're thinking about detrimental human health consequences?
I mean, the more contaminants we can eliminate, the better we
Of course.
are. and there are all interactions that we don't know about of PFAS with other contaminants. But if
Sure.
there are already indication from epidemiological studies, at least, that there is effect of humans, why do we need to increase in cancer risks? I mean, why do we need to delay development in the kids, right? That we already have the epidemic with developmental delays.
And so what you're
So I would say
what
that
I hear you saying is you're relying on that the epidemiological data is clear, right? So in other words, it's not just a correlation. It's they're confident from an epidemiological standpoint, and then they're finding plausible toxicological mechanisms. Is that how you would summarize it, or not?
that's pretty similar. I mean, if you have already evidence from human populations that there is adverse effects, I don't know if I want to question the effect at which they observed 4 parts per trillion is really, I did not see studies that says that they observed it at those concentrations. So it's really the immune response. I don't know if it's the right response to regulate it, but I know a lot of studies went into making that decision. So I guess they incorporated a lot of uncertainty factors, to be really, and that's the levels that assuming that you've been exposed over a lifetime consistently at this level, right? That eventually will result in those effects. So, but yeah. I mean, I wouldn't say we should stop and not chase. At least it's clear that, for example, the longer chains are definitely causing higher toxicity, right? Can we at least eliminate some things that are the most toxic? And then if you look at the different molecular weight, different aromaticity, right? Can we at least remove this? If it's possible, we'll decrease. But yeah, in the water, we have so much stuff, and we didn't even mention disinfection byproducts that also cause toxicity.
True. Yeah. Now, and so maybe 1 minute wrap up or final comments from each. Maybe we'll start with Shannon and then Olga and then Narapam. So Shannon, any comments to leave the audience with?
Yeah, I think just much more work is needed. I think even just on simple questions about occurrence. I think we have to also think about occurrence in categories of PFAS and not necessarily specific compounds, because this is a group of over 3,000 compounds. So we may use some of them as indicators or targets, but I think that's important to keep in mind. And then I think the other thing with occurrences, they seem, typically the paradigm is the chemical is released to the environment, and most of the time then it degrades. In the wastewater example that I showed, we actually saw potential, creation of PFAS within that environmental system. So I think we just have to think maybe about that there could be things we're doing to treat water or treat wastewater that potentially could result in higher levels of PFAS. So
Thanks, Shannon. Appreciate that. Olga?
The main message is safer sustainability, right? When developing treatment application, can we make sure that nothing is released and creating more problems and there's more interaction with PFAS? In our example is with nanomaterials, so that's why we're immobilizing it as much as we can or trying to figure out how to remove them when we treat. Right. And another thing that, there is so much unknown, so many PFAS, right? So that we just kind of scratching surfaces with them. It's impossible to evaluate toxicity of all of them. So I would bring Carla's back presentation from not the last week, but week before. Right, and she was talking a lot of modeling approaches, and if we can use it in combination with in vivo also toxicity, so for validation of the modeling, and maybe have some predictive mechanisms. So when we develop in technology, we can actually look at what it's breaking down and the potential toxicity instead of testing every possible breakdown product. So.
Thank you, Olga. Appreciate it. And Narapam, we'll go with some final words from you, my friend. Thanks again for putting this series together for us.
Yeah, no, thank you, Dan, for helping or even giving the idea that this is a very important webinar series for the community, and we are happy to help. Yeah, and I would like to thank Olga. It was not me only, it was Olga, Shannon, and Isabel, all of us together. So yeah, thank you for thank you to the whole team to make this happen. So my final words on the more on the treatment side, I think these are really great questions. Like we are, again, like Olga said, we are scratching the surface, and we are still talking about like individual treatment. Like we are looking at treatment of PFOA and PFOS, and when we mix them together, then they become complex. And when we mix other co-contaminants, they are becoming more complex. So we really need to think about like mixture treatment and like water chemistry, and the questions that came, like, can we think more from treatment train approach? I would say that. And I would like, overall, I have been in this area for quite a while, and I realized that people go after each other. Oh, I have this treatment technology. This is the best. And these others are crap. I think we should not do that. We should think about how we combine our efforts in different things. I don't think there is a single silver bullet. I think there is use for everything in some ways or another, and certain contaminant system or certain water matrices will need certain technology. In some cases, we will use this one. So I think instead of thinking from one treatment technology point, like thinking more from holistically, like, oh, this is our water quality. This is what the end use will be of this water. Can we use this technology to lower the cost? Or we don't need that pristine water in this use. And can we treatment have the treatment? Like, for example, the destruction technology is not needed everywhere. In a rural community where they cannot afford that much of cost, they may not need destruction technology. Then we can think about combining all of those separated PFAS and concentrated PFAS and bring it to somewhere else and do the destruction. So it's not like I'm designing destruction technology, so everyone should use destruction technology. So we need to think from that point too, like what the community needs. Like that's a major thing. Like we can develop as expensive as an efficient, but it may not work for the community that we are targeting to help.
Thank you. Thank all three of you. And Isabel, who's not here with us, thanks again. We'll turn it over to Marissa to wrap it up, but this has been a fantastic series. Thank you all so much. Marissa?
Yeah, thank you,
Okay,
Dan.
thanks
Have a
everyone
nice
for all
one.
your questions, and thank you to our speakers for sharing your knowledge with us today. We have several webinars planned in the near future. You can go to aaees.org/events and reserve your spot. Just a reminder, if you're not yet an AAES member and you're considering joining the Academy, you can email me to discuss our membership options. For all board certified individuals, PDH certificates for this event will be sent out by the end of the week. That's all for now. We enjoyed being with you all today. Thank you so much and have a great day.